Everyone must have noticed that humanity has taken another step towards the RoboCop. It can use a combination of human brain neurons and silicon chips for training and inference. Perhaps. Hello, welcome back to Dr. J Tech. I am Dr. J. I believe everyone has seen the movie The RoboCop. In this movie, RoboCop is a sentient being capable of cognitive thought, self-learning and evolving artificial intelligence humanoid robot. We have previously introduced the current artificial intelligence, which uses what is called a neural network. It is merely a mathematical algorithm, and the graphics processing unit, or GPU, is just a computer. So no matter how much a computer calculates, it will never have consciousness. It cannot think. However, if scientists were to extract neurons from the human brain and cultivate them on silicon chips, utilize these neurons to perform computations in the brain, could they create a computer with consciousness? Today, we will discuss this topic with everyone. So today's topic is the first generation of RoboCop becoming a reality. How does a real intelligent AI that combines human brain neurons with silicon chips actually work? First, we introduce to everyone the world's first biological computing platform debuts. Next, let's discuss the differences between human brain neurons and neural networks. Next, we present each CL1 bionic computer introduced now, equipped with 800,000 human brain neurons. Next, we introduce drug development on brain chips and more applications. The CL1 bionic computer is developed by Cortical labs, stemming from their early research efforts. DishBrain prototype for future technology. Next, let's talk about scaling biological computing from 100,000 to 1 billion neurons. Finally, we discuss the long-term goals, achieving human-level true intelligence. This is known as actual intelligence, commonly referred to as AI. Today, the source of our data is Shannon. Originally featured in IEEE Spectrum, the article explores the availability of human brain cells on a chip for purchase. World-first biocomputing platform hits the market. First, let's talk about the world's first biocomputing platform debut. Currently using graphics processing units, GPUs, or tensor processing units, TPUs, artificial intelligence, AI, that operates artificial neural network, ANN, models. This is what we call artificial intelligence, or AI. It's already becoming a mainstream topic, but using real human brain neurons fused with, instead of relying solely on the human brain, actual intelligence is achieved by using silicon chips to replicate real intelligence, also known as AI. Have you heard of it? It's like scenes from science fiction novels becoming reality. Australia's startup company Cortical Labs has launched the world's first deployable biocomputer is called CL1. This device was unveiled in March this year, integrating human brain neurons and silicon chips. It processes information through a sub-millisecond electrical feedback loop in under 1 millisecond, which is less than 1,000th of a second. The diagram below shows the internal structure of the CL1 bionic computer. On the left is the processor that integrates human brain neurons and silicon chips. Because it is built on real human brain neurons rather than purely digital models, so it has to be grown and maintained in a culture medium. Moreover, the lifespan of these human brain neuron stem cells is about 6 months. Next, we will introduce the human brain and the differences between neuron cells and human neural networks. The CL1 bionic computer is a new tool designed for neuroscience and biotechnology research, providing a fresh new option in the market, way to study how brain cells process and respond to stimuli. Unlike traditional silicon chip systems, this hybrid platform uses living human brain neurons. It can adapt and learn in real time and respond to external inputs. The word neural is related to the human nervous system. Neurons in the brain with a neuron visible on the left side. In the middle is the cell nucleus. The long part is the axon, and the end is the synapse. Human brain neurons are stimulated by chemical substances, causing the neuron to generate, which then travels through the axon and synapse, gets amplified and triggers the next neuron to generate its own signal, and it's amplified as it travels through the axon and synapse. So the human brain is built from individual neurons linked with each other to create a network of neurons interconnected in a neural network. The human brain contains approximately 86 billion neurons. In artificial intelligence, we often hear about artificial neural networks, ANN, also known as neural networks. It's a mathematical model designed to mimic the structure and function of biological neural networks. It's used to evaluate or approximate functions. The circles you see here represent neurons and the arrows represent synapses. Therefore, artificial neural networks are a model for computing probabilities. The so-called parameters of neural networks are weights, which are what we just mentioned. The amplification factor on the synapse of the neuron. So the amplification factor is the weight. Artificial neural networks have input layer neurons and hidden layer neurons, as well as output layer neurons. The circles and arrows here represent neurons, so a 1, a 2, a 3 are signal strengths, and W1, W2, W3 are amplification factors, which are the weights. When we input data into the artificial neural network through the input layer, after computation through the hidden layer, the probabilities are finally output by the output layer. Therefore, an artificial neural network is essentially a model for computing probabilities. When we hear scientists mention Facebook's Llama 3 large language model, having 400 billion parameters means having 400 billion weights, 400 billion magnification factors, 400 billion arrows. Here, both A and W are floating point numbers, which means they are numbers with decimal points. The so-called sequence involves converting images, text, and sound into floating point numbers, a 1, a 2, a 3, input into the neural network due to known probabilities. It is possible to repeatedly calculate what the 400 billion weights W are. This process is called training, and these 400 billion weights W are referred to as trained weights. Inference, as it is called, is using these 400 billion trained weights W to calculate probabilities and predict the future. When we incorporate the latest data, including images, videos, text, and sound, converting them into floating point numbers, a 1, a 2, a 3, input into the neural network through 400 billion training weights, we can calculate the probability of an event occurring. The University of London theoretical neuroscientist states that from a certain perspective, CL1, it can be seen as the first commercial biomimetic computer. It is a neuromorphic device, the ultimate expression of computing. It uses real neurons, which are brain cells found in the human body. However, the true value of this technology is not in computer science, but in the fact that it is a powerful experimental tool, allowing scientists to conduct research on a small synthetic brain. The first batch of 115 CL1 bionic computers will start shipping this summer, making it a mass-producible product. Each unit is priced at $35,000 but sold by the rack. If you purchase 30 units at once, the price per unit can be reduced to $20,000. Cortical Labs also provides a cloud-based option known as Wetware as a Service, abbreviated as WAS, for easy access and convenience. Each unit of Wetware as a Service is rented weekly with charges incurred on a per-unit basis of $300. You can access the neurons cultivated within the company via the cloud. Discussing individual units is important. The CL1 bionic computer is equipped with 800,000 human brain neurons. These cells are derived from real adult skin or blood samples that have been reprogrammed and cultivated in the lab. Cells can survive for up to 6 months through a life support system, maintaining nutritional supply, temperature control, and waste filtration, as well as physical balance. This means cultivating these neuron cells using a culture medium. During this period, these neurons continuously fire electrical signals, receive stimuli, and keep learning and adjusting over time. The CL1 bionic computer's energy and hardware requirements are extremely low, suitable for long-term experiments. An entire CL1 rack consumes only 850 to 1000 watts, far less than the tens of thousands of watts needed by NVIDIA for artificial intelligence computations. Data center configuration. The lead scientist at Cortical Labs overseeing research explains that brain cells generate tiny electrical pulses to communicate with larger neural networks. We can input similar electrical pulse signals to represent information bits and read their responses. CL1 bionic computer. All of this can be done in real time using simple code across multiple firmware and various hardware configurations and structures for computer systems. With sub-millisecond latency, meaning less than 1,000th of a second, can read, process, and write new information into the cells. This image shows what Cortical Labs has built. A processor that combines human brain neurons. The processor is surrounded by metal pins, along with silicon chips placed around it, are used for reading or writing electrical signals, and the tube above provides the culture medium. allowing the brain's neural cells to survive and perform computational tasks. Next, we will discuss brain drug development on chips and more applications with everyone. The chief scientist of Cortical Labs stated that they have received strong interest from universities, startups, and government agencies. The scope of applications includes development of new drugs, neural computing, and fast AI. In addition to mining Bitcoin, as well as from music and entertainment industry players have also expressed interest in collaboration. Think about it, everyone. If there were neurons similar to the human brain that could be trained to analyze, create, and produce music and entertainment works, is it possible for them to take over from humans? The company is currently exploring innovative applications combining biological computing and experimental art. For example, a recent art project originally intended to let CL1 and a deceased composer's brain cells interact with his brain cells. This means we can let the CL1 bionic computer interact with the neurons of the brain belonging to the composer who has passed away. This means that even after a composer has passed away, CL1 could still be used to create new music. Doesn't that sound unbelievably good? Cortical Labs positions the CL1 bionic computer as a foundational platform technology that can be used for epilepsy or Alzheimer's disease applications in drug discovery and disease modeling. At the same time, the chief scientist, it indicates that since we can use human brain cells to process information, that means we can use brain cells from different donors or different cell lines to identify potential disease-related genetic differences or even individual differences. He also noted that the failure rate of many neuropsychiatric drugs in clinical trials is extremely high. Part of the reason is that existing preclinical models cannot capture the true function of brain cells, which is how cells process real-time information. In fact, such a device is needed to conduct this type of model testing. You can first test the calculations, but this kind of calculation is actually an evaluation. It's a method to assess cell function and further observe whether it can be restored in diseases. Restore this function. In a recent paper on an in vitro model of epilepsy, researchers showed that the CL1 bionic computer successfully restored the function of damaged nerve cells. Epileptic cells originally could not effectively learn to play games. If anti-epileptic drugs are added to the cell culture, they suddenly learn better and exhibit a series of indicators that were previously unobservable. This image shows lab-grown neurons from Cortical Labs positioned above an electrode array. The gray part that everyone sees is the human brain neuron cells. The black part is the electrode array. Next, let's talk about how the CL1 biomimetic computer traces its origins back to Cortical Labs' original DishBrain prototype. CL1 is the result of further development built upon that initial DishBrain system. DishBrain has enhanced cell culture technology training to enhance its capabilities. To play a simplified version of a ping-pong game in a simulated environment, these cells learn to track the position of the ball and control the paddle, demonstrating goal-directed behavior even outside the body. In other words, the human neurons inside the CL1 biomimetic computer can be placed onto a silicon chip and run a game in a simulated environment, and learn to control the paddle by tracking the ball's position. According to a 2022 neuron study, these neurons demonstrated learning ability within minutes. They can self-organize and adjust behavior based on sensory feedback. In other experiments, Cortical Labs discovered these biological neural networks. In terms of sample efficiency and learning performance, it often outperforms deep reinforcement learning algorithms. DishBrain's proof of concept applied the free energy principle, proposed by Carl Friston, to test how the brain uses feedback mechanisms to minimize surprise in the active inference process, whether it can also appear in biological neural systems. Neuroscientist Carl Friston, the mastermind behind the theory, has detailed his description that the CL1 bionic computer is an astonishing achievement, a result of many years of theoretical and biophysical innovation. Friston also added that this allows researchers to study the effects of drugs and artificial lesions on neural circuits learning responses within a closed-loop system. When neural networks can interact bidirectionally with a simulated world, simply put, the experimenter now truly possesses a philosopher's dream. Decades of the brain in a vat, that is, a processor with the same functions as human brain neurons, and the scalability of biological computation can range from 100,000 to 1 billion neurons. CL1 represents a major expansion beyond DishBrain, including built-in life support system which allows human brain neurons to survive At the same time, the input channels increased from 8 to 59. Latency was cut from 5 milliseconds down to sub-millisecond levels, meaning less than 1 millisecond. Cortical Labs plans to further optimize CL1's performance and leverage the natural scalability of biological systems. This means that although the cost of producing 100,000 neurons is not low, but to produce 1 million or even 100 million neurons only requires a slight increase in cost, because biology grows exponentially. Only when you need to expand to billions or even trillions of neuron cells. It's like when cultivating lab-grown meat, you need to switch to other technologies. However, the scale of hundreds of millions of neuron cells is entirely manageable. Below is the operating interface of the CL1 bionic computer. On the left is the middle of the internal structure. This is the fusion of human brain neuron cells and the silicon chip processor, while these plastic bowls are for transporting culture medium, providing nutrients to create an environment where human brain neuron cells can survive. This is the CL1 bionic computer, which integrates a life support system. It allows neurons to survive for 6 months to perform computations. Although Cortical Labs does not intend to limit the application fields of the CL1 bionic computer, buyers need to obtain ethical review approval to generate cell lines. Customers purchasing the physical equipment must also have a suitable cell culture laboratory. Kagan also emphasized that they don't want this technology in the hands of people without proper expertise or safety controls. This is not garage-level technology. The young startup company Cortical Labs is making waves in the industry. Only 22 employees have already received support from around the world, with investments exceeding $11 million, including from Hong Kong's Horizons Ventures, Australia-based Blackbird Ventures, the CIA-funded venture capital firm In-Q-Tel in the United States. Cortical Labs is one of the dozens of companies in the life sciences investment portfolio, and their long-term goal is to achieve human-level actual intelligence in the future. Actual intelligence. Cortical Labs' long-term vision is to unlock human-level intelligence using human brain cells. Kagan defines so-called bioengineered intelligence as surpassing traditional petri dish methods. The brain's further stages ultimately aim to design a neural system capable of performing beyond human abilities. Kagan states that any advanced machine will eventually be indistinguishable from biological entities because we want machines to possess adaptability, aware, fix itself, save energy, and sustainability, which are all strengths of biological systems. With a sufficiently advanced collection of cells, you can achieve some accomplishments that might even surpass current biological capabilities, and such systems are also safer and more controllable than silicon-based chips. Finally, let's give everyone a simple conclusion. Through the introduction just now, everyone must have noticed that humanity has taken another step towards becoming the RoboCop. The ability to use a fusion of human brain neurons and silicon chip processors to conduct training and inference. Perhaps in 20 years, it might truly be possible to create a computer that is conscious and capable of thinking. At that time, will you start to feel afraid? That's all for today's program. For those who have questions about human brain neurons and bionic computers, feel free to comment below the video. Let's discuss again. Thank you everyone. Good night. Bye bye.